Welcome to the Ticktective podcast and video series, a program of the Bay Area Lyme Foundation, where our mission is to make Lyme disease easy to diagnose and simple to cure. I'm your host, Leah Gertner, Director of Education and Outreach. Lyme disease is the most common vector-borne disease in the USA and is a global issue. This show includes interviews with the researchers we fund, as well as other interesting people in the Lyme scientific community. We are a nonprofit foundation based in Silicon Valley. Thanks to a generous grant that covers all of our overhead, 100% of all donations go directly to support research and prevention programs. You can find out more or donate at bayarealyme.org. Welcome, Dr. Liz Horn, Principal Investigator for the Lyme Disease Biobank, a program of the Bay Area Lyme Foundation. Thank you so much for joining us on the Ticktective podcast.
Thanks for having me, Leah.
I know that our Lyme Disease Biobank has enrolled over 900 participants and supported over 50 research projects so far. And I'm wondering how this Lyme Disease Biobank got started. It seems really successful.
Yeah, we've been working on this for quite a long time. We actually started it in 2013, and our first season of collection was 2014. So it takes a long time to build a resource like this, but what we've really tried to do is to create a collection of samples from people, so human biological samples, to facilitate research in Lyme disease and other tick-borne infections, because we know that the diagnostics we currently have, they're not sufficient. And so we've tried to create a very broad sample collection to move better diagnostic tests forward. And that's why we have so many different researchers, both from academia and from industry, using our samples.
So what kinds of specimens does the Lyme Disease Biobank keep?
Most of our collection is blood and urine. And so once our participants consented to be a part of our research program, they give a blood donation and we ask them some questions about their medical history. So it's called a case report form, but it's really asking them about their signs and symptoms of Lyme disease, their medical history, any medications that they're on. And for our early Lyme disease study, we ask a lot of questions about exposure. So do you remember the tick bite? How long was the tick attached? Was the tick engorged? Did you have a rash? What did it look like? Was it red? Did you have central clearing? So we ask a lot of information to really focus on the signs and symptoms. So we collect blood and urine, and that blood is then processed to either whole blood or serum. And those are sort of the two main sample types that our researchers are interested in. So when we started this, we did a lot of interviews with scientists and we did surveys because we wanted to make sure we were collecting the right kinds of things that would help facilitate their research. So we had a group of advisors that helped us decide what to collect, the types of clinical history to ask about, and the questions we should ask our participants. And then we also collect a second draw. So for people that have signs and symptoms of early Lyme disease, we collect a second draw 2 to 3 months after the first draw. We also ask questions about their health. Do they have any ongoing signs and symptoms? Did they have any additional treatment? So in addition to our early Lyme disease, we're also collecting samples from patients who've had Lyme disease for longer periods of time, so that more persistent or chronic group of patients. And that's something that we just started in 2019. And so from our collection, we have several collection sites across the US. We have sites on the East Coast, in the upper Midwest, and also in California.
Great. And there's also a tissue collection that has begun with the Lyme Disease Biobank in collaboration with NDRI. Is that correct?
Yes. So NDRI is the National Disease Research Interchange, and they've been around since the late 70s. And their mission is to get tissue to researchers. And so they're not a biobank per se, but they collect tissue. And so we're partnering with them because I think that there are certain things that we'll only understand if we can actually look in tissue. So we can get tissue two different ways. The first way is we can collect samples from surgeries. So one of the areas that we're actually really interested in are knee replacements, because we hear a lot that people with Lyme disease have knee replacements. And one of the questions we have is it just osteoarthritis, or is there something going on in the knee with the Borrelia? And we can only answer that question if we can look at the tissues. So we have a program to collect samples from Lyme patients who are having knee and hip surgeries. And then we can also do other surgeries on a case-by-case basis. And so that's the first part of the tissue program. And then the second part is we have a post-mortem arm of the program where people who would like to donate tissues after their death can do that. And that's a process that people sign up for and answer some questionnaires. And it can either be the person or it can be their next of kin. And we've had a lot of interest in this. And we have what we call a living donor registry. And we have people of all ages that have expressed interest in donating their tissue. We know that the Borrelia goes deep into tissues. And so this would be a way to help understand it better. And then we could also understand Lyme disease in the context of other diseases. Is there a connection with Lyme disease and Alzheimer's? Is there a connection with Lyme and MS? And really teasing that out by being able to look at the tissue samples, both to look for evidence of infection, so evidence of the Borrelia being there, but also evidence of inflammation, so the actions of the immune system because the Borrelia is there. So we'd be able to answer a lot of questions if we were able to get tissue, but surgical tissues are really useful as well. So the only thing with that is the process is similar where individuals sign up ahead of time, they do an authorization, they fill out some questionnaires, and then NDRI works with the hospital or the surgery center to collect the sample. And one of the things that's really important with that is just having enough lead time prior to the surgery. So say it was an emergent surgery, like someone needed their appendix out, which we hope that doesn't happen to someone, but that would be really difficult for us to collect because it'd be very hard to arrange it with the hospital and the surgeon. Better for ones that are scheduled in advance.
That's good to know because I was just feeling really guilty because a friend who is a late stage Lyme patient just had her gallbladder removed yesterday. And I thought, oh, I should have told her, but she found out yesterday morning that it was going to happen. So she wouldn't have been eligible anyway.
And that's just so tough too, because then that's just, I'm sure, so much more stressful for the person to find out that morning, like, hey, your gallbladder has to come out.
Yeah.
So
how did you get started in the biobank world? I know you've worked with other biobanks previous to the Lyme disease biobank. How does one get to this place?
I had a very interesting path to get to this place. So when I was a graduate student, my advisor moved the lab from SUNY at Buffalo to Oregon Health and Science University. And as part of my project, I was a cancer biologist. I'm a cancer biologist by training. I had to set up a tissue bank because I needed head and neck tumor specimens and uninvolved tissue near the tumor for my research. And so we basically set up this project in the lab that has gone on to live and grow long after I've been gone, and it's still very active. And that gave me the background to set up a tissue bank. And then I was recruited to work for a nonprofit that was about 2 miles from my house. And one of the things we had to do there was to set up a blood bank and a research program. And so I spent my whole career really working with nonprofits and patient groups, building research programs, registries, and biobanks.
And so let's not forget there are also controls who are enrolled with the Lyme disease biobank.
Yeah, controls are really important in any disease because when you have a research question and you're looking in a specific patient population, you want to know that something is specific to the disease and not just different between different groups of people. So in early Lyme disease, we want endemic controls, so people living in the same areas that have the same potential exposure to Lyme disease but have never had any Lyme disease or tick-borne infections. So what we've done in the sites that we're enrolling people with early Lyme disease, we also enroll controls.
And you said you're taking late stage Lyme patients as well.
Yes, Justin. So that we're just doing in California right now. And we have 2 sites. One site is in Northern California in San Rafael, and the other site is in San Diego.
Along with some researchers at the Mayo Clinic, you published recently in the Journal of Clinical Microbiology. And the article was titled, The Lyme Disease Biobank, Characterization of 550 Patient and Control Samples from the East Coast and Upper Midwest of the United States. And that article had all kinds of pearls in it. The article states, a lack of provider awareness of this absence of erythema migrans or non-classical erythema migrans presentations can lead to underdiagnosis and delayed treatment, highlighting the need for better diagnostics for early Lyme disease. So I was just wondering if you could speak a little bit about that.
Sure. First, I just want to acknowledge all of my collaborators on the article. So that was 5 years of collection that we started in 2014 through 2018. And we had wonderful collaborators at East Hampton Family Medicine, in Martha's Vineyard, at the Marshfield Clinic Research Institute that helped us collect the samples. And then we had wonderful partners at Stony Brook University and Mayo Clinic to help us test the samples. And one of the things I think is really unique about the way we approached it is we enrolled people based on signs and symptoms of early Lyme disease. And one of those signs is the erythema migrans, the skin lesion, but not everybody has that. And so we also enrolled people where the provider thought that it was tick-borne that didn't have an EM. And so that gave us sort of two groups of patients, because we know that if you have an EM greater than 5 centimeters, that's classic presentation of Lyme disease. It's a clinical diagnosis. You don't need testing. But what about the people where their EM isn't quite 5 centimeters? They don't have any skin lesion. So we tried to have a broader inclusion criteria. And I forgot to mention New York Medical College. We also worked with them as involved with the sample testing. So it was really a great group of collaborators that helped us understand this collection. So one of the real challenges is that our current diagnostic, our standard two-tier assay doesn't work very well in early Lyme disease. And in our sample set, when we looked at people who had EMs greater than 5 centimeters, so that's a clinical diagnosis, only 29% of them were two-tier positive. And this can be really challenging because these are individuals that have the telltale lesion, but these EMs can all look differently. And a bullseye with central clearing is actually very rare. It's not how most of them present. It was almost 200 participants. So we took photographs of their skin lesions, and a lot of them are very red. They're large, expanding, annular, so oval-shaped lesions. Some of them did have central clearing, but very few of them really looked like that bullseye target. And so I think if providers and clinicians are really just expecting it to be a bullseye, that's not how it always appears. And sometimes they're fainter and they're not as red. So I think it's really important that providers in endemic areas are able to identify what an EM lesion looks like. And one of the things we talked about in the paper was that for people that don't present with an EM, it's almost impossible to diagnose them with our current tests.
I know John Alcott from Johns Hopkins has been talking about, I think the rate was about 20% of Lyme rashes actually look like the stereotypical bullseye rash, whereas all over in medical publications, you see that they say that it has to be a bullseye rash, and we're finding that that's just not true. But what you're also saying is that the two-tier blood tests, the results are not matching what you're seeing. So you're lucky enough to see an actual bullseye rash that came with a tick bite. That's diagnostic for Lyme disease. But what you're saying is that the blood tests were negative for some of those people where you absolutely know that they had Lyme disease, and the blood tests are 29% were positive, whereas 71% were false negative. Is that what you're saying?
Exactly. And I think the challenge is it's in early disease. So our blood tests really rely on your immune system coming up, and it has to detect antibodies. And that can take a while. And so with our clinical data, we asked a lot of questions about how long people had symptoms, if they had a tick bite, how long since they had the tick bite, to really try to get a sense of how long they were experiencing some of these signs and symptoms of early Lyme. And with our cohort, I think individuals came in sooner to the provider. We're also collecting in areas where people are very concerned about Lyme disease because they know how devastating Lyme disease can be and they don't want to get it. And it's just a very knowledgeable population. So they're coming into the provider and saying, hey, this is what I think is going on. And we're catching them earlier. And so the tests just don't work in early disease.
What were some other pearls that were highlighted in that study that you published this year?
There's two different types of antibody responses that you're looking for. Within a month, you're looking for your IgM, that immunoglobulin, to increase and go up. And then once you're out past a month, you expect to see IgG antibodies emerge. And so part of the diagnostic algorithm is understanding which class of antibodies you're looking at the time. And with our second draw, we did it at 2 to 3 months. And it was a little later than some of the other studies where they would look within 30 days. And we didn't see seroconversion. And that was unexpected. We had a very low rate of seroconversion, less than 5%. And Dr. Akot has published in his SLICE study that when individuals are treated with doxycycline, seroconversion is very rare. And the vast majority of the participants in the biobank have been treated with doxycycline. So that likely explains why we don't see seroconversion, but it also talks about the need for better diagnostics to be able to tell, especially in later Lyme disease, in someone that's been treated.
And what percentage of samples that you guys are providing to researchers are used for diagnostics versus therapeutic research?
I would probably say 90%. The vast majority right now is diagnostics. And I have to caveat that with most of the samples that have been made available are in that collection of 550 early samples. We've also created this really interesting, what we call the unblinded panel. So it's a set of samples of laboratory confirmed Lyme disease and controls with all the clinical data. And so it's 15 samples total, and you can test a new idea. If you're bringing a diagnostic from another field, if you haven't tested in a lot of human samples, you can test in this panel. And usually when you test blinded because you don't want to be biased. You don't want to know which ones are the cases and which ones are the controls. But when you're very early on, you just want to get an idea. Might your assay work in this space? What areas do you need to work on? Where do you need to refine it? So the unblinded panel really helps groups that are early in their process, and it also gets them some preliminary data to then be able to come back and ask for additional samples. Because most, sample users that are getting several hundred samples are much further along in their diagnostic process than the groups that are just getting smaller amounts. So that's been really fun to be able to help researchers who are newer to the Lyme field.
So I know at the Lyme Biobank, you guys did the 2-tier antibody testing on the Lyme disease samples. Was there any other kind of testing performed, like PCR testing on these human samples?
Yes, we did PCR testing on all the samples for both Borrelia burgdorferi as well as Borrelia miyamotoi. And then we also checked for Babesia and Anaplasma. We were looking at these other tick-borne infections because they're pretty common in the places that we were collecting samples. So Babesia is pretty common on Long Island, and Anaplasma is pretty common in the upper Midwest. So one of the things that was really interesting is we got very few Borrelia positive PCRs. It was less than 5 out of the whole collection. And those that were positive for PCR, so we just did PCR of the blood, so the Borrelia still had to be in the blood. And we know that the Borrelia likes to leave the blood and go in the tissues. So you have to catch that right window. So one of the things that was really interesting about the samples that were positive by PCR, they were negative by antibody testing. So, and others have seen this as well, that when you're able to detect the Borrelia, it's earlier in the timing than when you can detect the immune response. And really, to have better diagnostics, we need to be able to have a way to detect directly, to look at the pathogen, as well as looking at the immune response.
So hopefully some of the diagnostic research that is making use of the Lyme disease biobank will help come up with a direct detection test to accompany perhaps the existing antibody-based tests that we currently use.
Yes, and that's one of the main reasons why we're collecting the whole blood, because usually direct detection is often a PCR test.
Why don't they do PCR more often?
It's really insensitive, and it takes a lot of resources, and the reagents are expensive. And if you look at all the cases we tested, we had such a small number that were positive. One of the things that was kind of interesting about how we did this study is we tested all of the samples, even all of the controls. And so one of the things that's been interesting is that we do have some controls that had some background positive serology on one of the tiers. And it could be a false positive. It could be someone that perhaps was exposed to Lyme disease, but never really thought that they had Lyme disease because they weren't sick. And so that's this one part of the sample collection that's been really interesting to researchers, because we actually have controls that have some positivity. So that's important in testing their assays as well.
Right. I know there have been studies in the past that have shown that a significant number of controls will test positive, and we don't know whether they're asymptomatic individuals or whether it's a consequence of testing being imperfect.
Right.
Now that we have coronavirus, more and more people in the mainstream world are understanding the complexities of diagnostics that you have been dealing with for years, and people in the Lyme disease research community have been dealing with for years. So it's definitely an interesting time.
And one of the things I'm actually really hopeful is it will generate more awareness about how important accurate diagnostic tests are. So you need both the direct detection to know if the pathogen is there, and then you also need the antibody response to understand what's happening at human levels. So all of these articles and issues with COVID-19, they're very similar to what's happening in Lyme Diagnostics. And if you don't have a good test, it's a real problem because you need to know if you need to treat or not. And so I'm hopeful that this will help generate more awareness. And then the other thing that it's doing is a lot of Lyme researchers, because they are, they're diagnostics people, they're all working on COVID tests. And I think they're learning a lot. We're putting so much emphasis on COVID, which we should, because this is unprecedented and it's really important, but I'm hoping some of those learnings will then translate back to Lyme. And when we're working on better Lyme diagnostics, that we're further along than we were last year.
I know it's been sad to see so many of our Lyme disease researchers have to put their Lyme disease research on hold, either because they can't go into the lab because the labs are shut down, or because they've had to focus on coronavirus research. But I like your way of thinking about it, that this could help further the Lyme disease research.
So I talked to a Lyme disease researcher today, and he was so excited because they had taken a COVID test from soup to nuts in the lab, and they showed that they actually could do it. And they're like, oh, so now that all of that learning, all of those pieces, that's going to translate to Lyme. So I think that while we're all disappointed that maybe things have slowed down a little bit in Lyme disease, that we'll be able to move faster. But it's definitely been interesting times.
Thank you, Dr. Horn, for joining us today. And thanks again for all the hard work that you're doing to support the Lyme research community.
Thank you so much for having me. And before I go, I really just wanted to thank the Bay Area Lyme Foundation for really believing in this project, for supporting it all these years. And I'd also like to thank the Stephen and Alexandra Cohen Foundation for supporting this project, as well as all of our research participants who have been so generous to be part of this study, and all of our collaborators, because this really takes a village. And we're just so grateful that there are so many people working on Lyme disease and trying to make things better for people who are suffering.
Amen. Well, thank you, and stay healthy.
Thanks, you too.
Thank you for joining us for this episode of the Ticktective podcast and video series, a program of the Bay Area Lyme Foundation. You can find out more or donate at bayarealyme.org.